Hello everyone, welcome to BrainMap. This seminar series is co-sponsored by the P41 funded Mesoscale Mapping housed in the Martinos Center. It is our great pleasure to have Dr. Jetro Tuulari from University of Turku for our BrainMap talk today. Dr. Jetro Tuulari is a neuroscientist and an expert on multimodal MRI and interdisciplinary research of developmental cognitive neuroscience. He and his research group study how early life exposures, genes, and environment shape the brain structures and functions, and how these integrational factors relate to health and well-being. His research team is based on in the University of Turku, Finland. He is currently principal investigator of Fin Brain Neuroimaging Lab, part of Fin Brain first cohort study, and director of Turku Brain and Mind Center, and the head of neuroimaging in the Center of Excellence for Learning Dynamics and Intervention Research. I would like to remind our audience to address your question into the Q&A box or by raising their hand. Thank you very much for coming today, Dr. Yetru Trari, and the stage is yours.
Thank you, first of all, for the kind introduction and then also for inviting me. This topic has been at the heart of my research activities for about 8 years now. I'm very happy to share insights with you today. First of all, I come from the University of Turku, and for those of you who don't know where Turku is, it's located here in southwest Finland. And I'm based in FinBrain birth cohort study that was started in 2010. And this cohort is a prospective cohort that started measurements from gestational week 14. So after the first trimester of pregnancy, it's been led by Hasse and Linnea Carlson, and it has grown quite large over the past decade. One of the core research questions of the FinBrain study is how maternal health during pregnancy influences child brain development and behavior. The contents of the talk today will include one of the core hypotheses, introduction of one of the core hypotheses that these studies are based on, called developmental origins of health and disease hypothesis, a short introduction on prenatal maternal distress and how studies typically measure that, very brief overview on prenatal brain development. Then I will also give an overview on maternal prenatal distress and brain development, how these two have been linked in studies so far, touch the importance of early postnatal environment, and conclude end with conclusions and future directions. Now, of course, when we are talking about links from maternal well-being to child brain development, the whole concept is based very much on seminal findings from David Parker from 1989. And his most important finding was that low birth weight and low weight in infancy increased the risk of death from ischemic heart disease. He later proposed a hypothesis that is commonly referred with his name. The Parker hypothesis proposes that fetal and early infant conditions have a permanent conditioning effect on the body's metabolism and chronic conditions later in life. Of course, this specifically refers to metabolic health, and similar hypothesis is nowadays commonly referred under the developmental origins of health and disease, or the DOHAD hypothesis. Basically with the same idea that prenatal and early life, so including early postnatal exposures, can affect later health, and multiple types of exposures, including maternal prenatal distress, can be grouped under the DOHAD hypothesis. I think it's important to note that for interpreting the findings that I present and in this field, It is important to note that the exposed individuals are not necessarily programmed for later disease, but many exposures, including maternal prenatal distress, can alter brain development and increase later disease risk during that individual's lifetime. The environment, lifestyle, and health-related behavior will always affect the health outcomes. And then lastly, many of these studies are epidemiological in a way that they describe associations across a group of participants and are not meant for predicting individual outcomes. Prenatal maternal distress is fairly common. Studies report that it affects 10 to 35% of all pregnant women. And we also know that maternal prenatal distress associates with slightly increased rate of perinatal complications. You might take note that the range of the prevalence, 10 to 35%, is quite large, and this is very much related to the way we quantify and measure stress and distress. And I acknowledge that stress regulation system and stress system in the human body would easily be a topic for a 1-hour lecture on its own, but just one figure on this topic. So here is plotted stress levels on the x-axis and physical activity on the y-axis. And there's always a level of stress that is suitable for an individual. It's commonly called eustress or good stress. And actually, if we have too little stress, it tends to be poor for our well-being. And if we have increased level of stress, we start experiencing distress. And then when the stress levels have increased to distress levels, we experience symptoms like fatigue, exhaustion, anxiety, and get also adverse health effects. Now, in the studies that have quantified and measured maternal prenatal distress, typical proxies or measurements for stress are based on questionnaires. They typically, and the questionnaires can quantify many different things: depressive symptoms, anxiety symptoms, perceived stress levels, or then ask about stressful life events that are commonly thought to be stressful, in addition to the symptoms that the participants are experiencing. Now, of course, feelings of stress will not directly transfer from pregnant mothers to their developing fetus. And I'm going to introduce you two key physiological systems that are thought to mediate or convey the effects from mother's physiology into the fetus. The first one is called HPA axis, standing for its components. Hypothalamus and pituitary in the brain secrete hormones that then cause release of cortisol from adrenal glands. Cortisol is a very important stress hormone, and it is known that it can also influence fetal physiology. The other mediator or core system that could be causing these effects is immune system. And I must say, even though this graph looks very complicated for immune system, it's fairly simple one. It's very intricate in the way the cortisol system and the immune system are interlinked. The main message perhaps in this PC figure is that stress and depression can cause changes in brain balance that again are linked to the HPA axis and cortisol levels, and then to the immune system. And these systems are interlinked and able to convey cytokines and other molecules that then produce the final effects. So there's a bilateral communication from the stress regulation system into the brain, and the cortisol system and immune systems are very important in this response. Of course, then how all of this can affect fetal brain development comes, of course, through the placenta, which is the communicating organ in between maternal and the fetal circulation and also physiology. And of course, prenatal exposures can come from changes in maternal physiology, or they can be external factors that come through maternal bloodstream. And these could, for example, include alcohol or other medication or other molecules that are circulating in maternal bloodstream. Placenta is a very key structure, and it's also a safeguard for many of the physiological and also external substances that are circulating in the maternal side. The defenses, in quotation marks, include also safeguards for cortisol levels and immune system, but there are also physiological changes that in highly stressful situations that some of the placental safeguards might be downregulated so that the cortisol and immune system can affect the developing brain. One key motivation for studies addressing prenatal brain development and maternal distress comes from epidemiological studies that have linked prenatal distress with autism spectrum disorder, increased risk for ADHD, conduct disorder, and depressive and anxiety symptoms later in life. And of course, this brief list includes many conditions that are thought to be brain-related. And then this has provided a key motivation for neuroimaging studies to look into neural correlates of maternal prenatal distress in during early life and especially in neonate populations. Prenatal brain development is very fast, and I would like to start with the fact that in terms of cellular processes and steps, general steps in brain formation, it's highly conserved across species. So you find the same basic steps in mice and in humans. And these basic steps include neurogenesis that happens during pregnancy, meaning birth or creation of neurons, synaptogenesis, meaning the forming of connections in between neurons. It starts during prenatal period and continues rapidly right after birth, and then myelination. And the myelination improves the connections and communication between the neurons throughout the brain. And of course, what separates humans and mice, for example, is the total brain size and the complexity of the brain tissue. And one noteworthy thing for humans is that even though the steps are kind of similar as in smaller mammals, we do see that they are prolonged up to 15 years and 20 and 30 years. And the peak brain growth happens around birth, so right before and right after birth. And key concept for the developmental origins of health and disease hypothesis is that these creation and developmental steps are the most vulnerable to external exposures as well. The whole embryological development of an individual is, of course, very marvelous on its own right, where by the end of gestational week 7, most of the organ systems or their forms will have taken shape. And by the end of first trimester, by gestational week 14, which is depicted here, the brain and most of these parts are already formed, but the brain tissue is still flat. And by mid-gestation, gestational week 26, the basic shape is even more readily developed, but the cortex is still flat. And after that, one of the major processes that continues throughout the end of pregnancy is cortical folding. And the cortex folds, as the cortex folds, it's able to accommodate more surface area in the same volume. And this basic step happens in many other organs, including intestinal systems and lungs. But of course, this folding forms the sulci and gyri in the brain and also gives it an adult-like appearance. And actually, by term age, many of the major cortical anatomical landmarks are already present in the neonatal brain. So it is already, in many regards, similar to an adult brain. One of the key differences, of course, is brain size. It's not the size of the adult brain. And just to give you an idea on the volumetric changes or the growth in brain size during pregnancy. So mid-pregnancy would be roughly here, around 24 weeks of gestation. The brain volume is roughly 200 milliliters or 6.7 ounces. I do hope I got the conversion right. We use the metric system here in Finland. And then from that 200 milliliters, there's a growth of basically a doubling by term age up to 400 milliliters or 13.5 ounces. So that's a very large volumetric growth. And then on the right-hand side, there's plotted, this would be mid-gestation, so halfway through the pregnancy, and this would be equivalent to term age around 37 weeks. And you can appreciate both the growth in size and also the change in shape from mid-gestation and basically flat outline of the cortical mantle into more complicated and more adult-like pattern by term age. These figures have been taken from an article from last year where investigators were able to create brain charts or brain growth charts for the human lifespan, basically mapping normative brain sizes across the human lifespan. Many of us would know growth charts from child healthcare and health screening, where there are physicians review charts on height and weight and whether or not they fit the normal ranges. And this is something similar now done to the human brain. The median growth chart is depicted in red for females and then in blue for males. And these are now for the cross-tissue compartments. So gray matter is depicted here in purple, and white matter is depicted in blue, and then subcortical gray matter in yellow. So these are the three main tissue compartments that the brain growth charts were created for. This is, of course, very important information on normal development. It can be used in studies, but maybe the main point here that I would like to point out that for each of these tissue compartments, before birth and up to age of 2 years, more than 50% of the brain size will be achieved for all of the major tissue compartments by the age of 2 years. So it is really brain development and brain growth during prenatal period and early postnatal period is very fast. Then to an overview of maternal prenatal distress and brain development. I'm focusing on studies that have been done with MRI imaging. Here are a few photos from our own data collection carried out as part of FinBrain birth cohort study. On the left-hand side, you see an infant that is soundly asleep after feeding, and it's been prepared. So we use this vacuum mattress so that the baby is tucked in tight and is keeping warm. And then our radiographer is placing the infant to be ready for the scans. Many studies on this field have been done in neonatal populations. it is maybe surprisingly one of the easiest ages to perform imaging on, because infants spend a lot of time sleeping, and whilst they are sleeping, we are able to then do MRI imaging quite successfully. This is not quite the case for example, 2-year-olds that are very, very challenging to image. So neonatal populations will be one of the most common populations in these type of studies. The other nice thing about imaging neonates is that postnatal life has been so the postnatal exposures have not been there for an extended period of time. So it is one of the best time windows to image prenatal exposures and their influences on the brain. There are, of course, many types of exposures that can affect prenatal brain development. This list is taken from one of the reviews that we've carried out in our research group, and we identified many different types of exposures. These include environmental toxins, for example, lead exposure, maternal substance abuse, tobacco smoking, alcohol abuse or drug abuse, some medication, for example, antidepressive medication. And of course, these three would be external chemical exposures and not part of natural maternal physiology. And then we also identified studies that addressed maternal obesity and nutrition, maternal mental health, including depression, and then maternal prenatal distress as one of the key exposures. And these three are then linked to the fetal brain development through maternal physiology. As a summary, I thought I would give main conclusions from a review article written by Alexandra Lautarescu and colleagues. This is from 2020, so from 3 years ago. They identified multiple studies that addressed effects on fetal brain and child development from maternal prenatal distress. The number of studies they identified were 25 structural MRI studies, 19 diffusion tensor imaging studies, and these are studies focusing on the structure of the white matter or the connecting tracks within the brain, and then 5 fMRI studies. Now, of course, there have been quite a few studies after this review published on this area, but I think the main conclusions of this review are still quite accurate. They conclude that infants exposed to higher prenatal stress have larger amygdala volumes, disrupted white matter connectivity between the amygdala and prefrontal cortex, and functional connectivity changes between these two structures, so amygdala and prefrontal cortex. They also conclude that these cerebral or brain changes could plausibly underlie many of the emotional, behavioral, and cognitive changes that have been linked to prenatal stress. There are, of course, many limitations into these conclusions. Many of the studies, so the amount of studies is fairly low. Many of the studies have quantified stress exposure differently, which kind of hampers all many of the comparisons we can do in between the studies, and only part of the studies have actually measured biological samples that would capture the cortisol system or immune reactions. And there are also many discrepancies in between the studies, but the most replicable findings are enlarged amygdala or larger positive association in between distress and amygdala size, and then connections, lower quality of connections from amygdala to prefrontal cortex. And here on the figure on the left-hand side, you see amygdala, left amygdala in red and right amygdala in yellow. Amygdala is located in the central parts of the brain and is very crucial brain structure for emotions, including negative emotions. And then the other key player, prefrontal cortex, is basically located here. And the key finding is that larger amygdala and connections from amygdala to the prefrontal areas are linked to prenatal stress exposure. And I suppose one could think this as the amygdala being part of the limbic or the old brain, sort of an engine room of the human brain, and then the prefrontal cortex would be the main control center of the brain. And of course, imbalance or changes in these two key structures have been associated with many brain-based conditions like depression and addictions later in life. I really like this two-level thinking. This is captured from one of our leaflets for the families, but it gives like a nice idea on there when there needs to be a balance between the engine room and the control room, or the amygdala and the prefrontal cortex. And again, maternal prenatal distress could influence the way these areas are growing and then wired to one another. Then I'm going to give you one example from our own studies where we mapped both sides of birth, meaning prenatal and postnatal maternal distress. We did this in 5-year-old children, and this is, I saw you had previous section in BrainMap for pediatric MRI and its challenges, but of course, it's fairly easy to imagine that it can be fairly intimidating for the children to approach and then go into the MRI machine. Here's a photo taken from one of our data collections that we carried out in Turku. And we spent a lot of time preparing the children. For example, they could place their head in this wooden replica of the head goal of the MRI machine. They were able to MRI image their own favorite toy before going into the scanner room. And we spent a lot of time preparing and assuring that the visits were safe and nice for the children. So family-friendly MRI visits. And in this data collection, we were able to get 130 children to take part in diffusion tensor imaging measurements. And diffusion tensor imaging is commonly used to image the white matter in the brain, so the connecting tracks in between brain areas. And in this study, we used derived brain measure called FA, fractional anisotropy, which is giving an index of coherence or the uniformity within the tracks. So kind of the higher the FA value, the higher the tissue organization in that brain area. And we were specifically interested in looking at both pre and postnatal maternal distress, and we used depressive symptoms and anxiety symptoms in this study, but found most of the results only for the depressive symptoms. One key reason for this is that myelination, which is key step in kind of effective, one of the key steps around birth, it starts to accelerate from mid-pregnancy and then continues to be rapid soon after birth. So that's where we were interested in white matter structure specifically in this study. Again, imaging that, so we had the exposure during the prenatal or perinatal period, and then the neuroimaging at age 5. And we found different results for males and females. Here are the results for males, where we find that prenatal second trimester EPDS scores, so Edinburgh postnatal depressive scores, were positively associated with fractional anisotropy after controlling for EPDS scores 3 months postpartum. And we also statistically adjusted for many of potential confounders that are known to or could influence brain development. And we found this negative association in the middle parts and in the posterior parts of the brain. For females, however, we found an association from postnatal scores, and the depressive symptom scores at 3 months postpartum associated negatively with fractional anisotropy in widespread areas after controlling for the prenatal distress scores. And we did capture some of the same areas than in males or boys, but then also these frontal brain areas. And this finding is very well in line with prior literature showing prenatal distress associating with lower FA values in the frontal areas. Now, the novel thing about this study was that we found these associations to specifically to 3 months postnatal, so after birth. And then from this study, we conclude that prenatal and postnatal maternal distress is associated with brain white matter development, and these associations are sex and timing dependent. And of course, future studies are needed that also measure behavioral data so that we can define the relevance of these associations to child outcomes. We did not include any behavioral outcomes into this study. I find the results that we got that some of the associations were sex specific, and also that the timing pre and postnatal were different, very intriguing. And there are a few studies reporting similar findings. And the problem here again is that if prior studies have not directly tested this type of hypotheses, it's very hard to know. So we would need kind of replication of older data sets probably also to advance knowledge on this topic. And of course, pre and postnatal exposures will have also different mechanisms. We covered how depression, anxiety, and distress can alter gestational biology, cortisol system or the HPA axis, and immune-related pathways that can then alter infant brain development through maternal physiology. And of course, after birth, one of the main exposures that the child is then experiencing is caretaking. And this figure is taken from a review article that specifically addresses postnatal environments that promote resilience in relation to prenatal stress. And they identify a few things that are known to promote or buffer any prenatal exposures. And these include cognitive stimulation, linguistic stimulation, better parental caregiving quality, and then distal factors like social support and higher socioeconomic status. So both timings before and after birth are important, but have different mechanisms. This topic has not been too much investigated. This figure is taken from Ruth Feldman's article. Her group has pioneered studies on behavior or synchrony in between the infants and children and their caretakers. And they're calling it biobehavioral synchrony, meaning that the synchrony is manifested both at the level of behavior and also at the level of physiology. And it is very interesting to see that this type of supporting parenting and caretaking quality can have positive effects on later brain development. So even though the individual's genetics and prenatal exposures have a lot of programming effects on the brain, many of the outcomes will be affected by the parental caregiving behaviors and quality. Then to conclusions and future directions. The research that me and my group are carrying out and many similar studies are basic research in a way that we are mapping out how subtle changes in maternal health, often subtle changes in maternal health during pregnancy influence the developing brain. But I was very happy to see when I was searching online for this topic, many resources that frame maternal well-being during pregnancies to also include thinking about baby's brain development. You can find many good things like maintaining a healthy diet, increasing exercise, refraining from smoking and drinking alcohol, managing your stress, and avoid exposures to environmental toxins and airborne toxins. So many of the things we identified as being influential to prenatal brain development are listed in these instructions already. The other nice thing is that this also seems to be now part of policy recommendations. I was very happy to read one article from last year that gives policy insights with the baby brain in mind. The recommendation states that maternal prenatal stress is preventable, treatable, and tractable by policy, and they give 5 different policy recommendations, including increased screening, encouraging talking about prenatal mental health, introducing evidence-based interventions for pregnant women, and avoiding stress-inducing communication, and lastly, stimulating positive postnatal parenting. Now, I mentioned that this is fundamental or basic research onto the brain development, and to advance the field, I think we have to move back in time, so kind of not back in time, to include both sides of birth, so including prenatal and postnatal, early postnatal development. And also, one of the key challenges for the field is to increase the sample sizes so we get more certainty on all of the associations. There are many nice open access data sets and consortia on the way. These include the Developing Human Connectome project that has imaged over 1,000 fetal and neonatal MRI scans. The Baby Human Connectome project, which includes around 500 participants from ages from 0 to 5 years of age, and then Healthy Brain and Child Development Study that has been recently launched. It will include participants from birth to 10 years of age into longitudinal measurements of behavior, biological samples, and neuroimaging. So these wonderful data sets that can be made open access to the scientific community will be a very important resource for neuroscientists in this field. I feel important to mention Enigma Origins Consortium, which was fairly recently established. It's run by Rebecca Knickmeyer. It's called Organization for Imaging Genomics in Infancy, and this is specifically tailored, or one of their goals is to collect large data sets that have both genetic data, neuroimaging data, and behavioral data. And these types of large data resources will be also very valuable to the future. So I'm guessing the coming 5 to 10 years will give a lot more information on this topic. Finally, collecting, analyzing, and processing neonatal brain scans and neuroimaging data from young participants is often technically challenging, and recently there has been established a new group called Fetal, Infant, and Toddler Neuroimaging Group. So if any one of the listeners are interested in learning more about imaging done in these young age groups, I highly recommend finding their web pages and Twitter feeds and other resources they provide. Lastly, I would like to thank all the wonderful people that I have had the pleasure to work with over the past few years, including many of the members in our team now. Yeah, thank you for your attention.